Thank you for this invitation. And today I will present how the Neuropixels Opto probe allows us to activate and record from local neuron populations in the mouse visual cortex. And this work is a part of the Neuropixels Opto consortium, and I have performed experiments while I was a postdoc in Matteo Scarandini lab at UCL. And I will start with this general question. Key question in system neuroscience is how computations performed locally within cortical circuits give rise to distributed processing across brain regions. And the cortex has this layered structure where each layer has its own inputs and outputs and local connections that together shape how information is processed. And to really understand how these local circuits process information, we need to go beyond just recording activity and use a tool that will allow us to both record and casually manipulate neurons. And that will be particularly important to identify which cell types we are recording from and are active, manipulate neurons in defined location, and also measure how those manipulations affect activity across layers and general the output of the cortex. And as Nick already introduced, this is a new prototype of Neuropixels probes that allows for combining both optical manipulation and electrophysiology recordings. And I'm not, since Nick already talked about some specifics of these probes, I'm not going to go into details, but I wanted to highlight that this is a first prototype. And here the light that has been delivered to the probe was delivered outside of the probe through a fiber cable and was coupled into on-chip waveguides that terminated in these tiny emitters positioned near recording sites. And in these probes, we could open one emitter at the time, so we could deliver the blue or red light just from one emitter. And one of the important part was also to cover those probes with material that will reduce some light artifacts and also increase mechanical stability during the probe insertion. And here I'm showing how the optical characterization look like for those probes. So as I mentioned, the probes had blue and red light coming from one of these 14 emitters. And this is a top view, front view, and side view and front view of the light coming from one of the emitter, which was measured in water. And what you can see is that this light, the light generated through emitters was narrow, were confined beams that extended roughly around 100 micrometer from the shank. And if we look at the coverage along the recording axis, you can see that these are overlapping illumination zones, which is important to making sure that we stimulate neurons in each recording site with high spatial precision. So with those probes, we can actually address those questions in this big question that I pointed at the beginning by trying to manipulate activity in the cortex and therefore directly link the structure and function of neurons or specific layers in the cortex and understand better how those manipulations or local stimulation shapes network dynamics across layers. So today, I'm going just to focus on one part of using these probes and testing those in the visual cortex, and I'm going to focus on how we use these probes to locally activate neurons and study effects that propagated through the cortical volume. So To do that, we tested those probes in the mouse visual cortex. In these experiments, I used only red emitters, and the goal was to activate CAMK2 expressing neurons that are putative excitatory neurons at different cortical depths using this redshift and very sensitive opsin, and estimate the spatial precision of this activation. Animal was head fixed, and we used different protocols to manipulate and stimulate activity. The pulses were randomized across 14 emitters, red emitters, randomly interleaved with control trials like external illumination and visual stimulation, and repeated 40 times. So in this first question that we asked was to see whether this probes may induce some light artifacts. And we implemented several strategies to reduce these photoelectric artifacts. The first was to use the light pulse that had a smooth onset. And the second was to apply a high-pass filtering, phase shift correction, and common median subtraction prior to spike sorting. And when we look at the raw recordings after application of those two, what you can see here, these are a single channels recording the activity of neurons in the visual cortex with a zoom on the location where the emitter 11 was on neurons close to emitter 11. And what you can see is that this huge spiking activity, it's not necessarily an artifact because it's very localized. And we could actually successfully reduce the light artifact by applying this too. And here we can see example of free waveforms from free units that were recorded near the emitter 11 in this case. And then we wanted to make sure that the spiking shape is not affected during the light stimulation from the emitter. And for this, we compared the shape of waveforms during the baseline period without optical stimulation, during emitter 11 stimulation and external surface illumination. And what you can observe here from these three examples is that the waveforms remain stable. They had very similar amplitude and shape across all of these three conditions, meaning that the light pulses from emitter didn't distort the electrical signals. And next, we wanted to identify laminar boundaries in these recordings, and to identify the position of cortical layer 4, we use current source density analysis, and that allow us to estimate the position of layers in relation to the position of emitters. And here, what you can see, this is example of activity. So basically, with current source density, we can see we can follow visual stimulation and see areas current sink shown here in blue that corresponds to putative layer 4, and based on this estimate the position of other layers and see how how stimulation on the probe induced the activity in the visual cortex. And this is an example of the stimulation during from emitter 11 in this example, and you can see that there's some small activity, and when we look at the activity triggered by single others emitters, you can see that this activity propagates across cortical depth. And this is not quantified, but it gives you just a sense that possibly with one emitter, we could induce manipulate the activity in layer 4. But this method just allows us to align the depth of each cortical emitter precisely along a cortical column and see if we can use a single emitter to manipulate this activity. So next, we show that emitter along the probe drives neurons locally, corresponding to the depth. So here we focus on a average spiking activity, and first we confirm visual responsiveness across cortical depths, and the strongest activation occur in the superficial cortical layers, verifying that the probe was placed in the visual cortex. And this is the average of multi-units and putative neurons along the depth of the cortex. And when we look at the impact of optical stimulation coming from a single emitter, you can see that this activity that was triggered by single emitter nicely goes along the depth, meaning that these activation maps are tightly confined and repeatable across sessions, showing that the probe can stimulate distinct cortical depths. And this is that was one example, and we could repeat this in 3 different animals, so the pattern is consistent. And on average, activation spreads about 150 microns, showing that, but that depends, of course, on the expression of the individual animal. And just lastly, this is just to prove that what we show here, it's not artifact. And I'm showing here that animals without opsin, the light stimulation didn't evoke any activity, and confirm that these observed responses were truly optogenetic and not thermal or photoelectric artifacts. And to summarize, these probes enable simultaneous recordings and precise optical control of neurons. They drive localized activation of cortical populations without thermal or optical artifacts. And the probe provides here high spatial and temporal control. And of course, this is just a proof of concept, and you can think about using these probes in the future for local and multi-region neural circuit manipulation, some closed-loop experiments, or optotagging and functional mapping during behavior. And with this, I wanted to thank UCL team, particularly Matteo Carandini and Michael Krumin, and also Neuropixels Opto Consortium and our funders. And thank you for your attention.